Hello, everybody. I'm Giulio from Ca' Foscari University of Venice, and I will introduce in this talk a novel Chimera dictionary data structure based on sparse and skew hashing. So we'll start off with a bit of context and motivation. So we'll know very well that public repositories such as ENA, SRA, RefSec, Ensemble, and so on contain billions of assembled sequences. So these collections are, of course, very important since they are paving the way to answer fundamental questions regarding biology and entire evolution. But before being able to have a look at the data at this scale, we need, of course, efficient methods to index and search such collections. And so one popular strategy to handle such complexity is to reduce a DNA sequence to a set of short substrings of fixed length k that are called the k-mers. And here, you have a simple example for k equal 13. So as a result of this, software tools based on k-mers are predominant in bioinformatics, also because k-mers are relevant for many different applications, ranging from genome assembly, variant calling, to sequence comparisons and alignment. And most of such applications need to maintain actually a set of k-mers and test membership to such set, which brings me to the specific problem we consider in this work, which is the so-called Chimera dictionary problem. So we are given a large DNA string, which can be, for example, a genome of the full genome of an organism or a pangenome, and let capital K be the set of all its n distant k-mers. So the problem we consider is that we want to build an efficient dictionary for this set, capital K, so that the following operations are efficient. So we want to do a lookup operation for a Chimera G, which returns I, which is a unique integer identifier between 0 and n-1 if the Chimera G is actually present in our set, or I is equal to a default value, say, for example, -1 otherwise. So if it's not present in the dictionary. And then we also want to do the access operation, which takes as input an integer I in between 0 and n-1 and returns the corresponding Chimera string G. So that's the inverse of the lookup. I will focus in this talk on the lookup query since it's the core of the whole data structure, and can also be used to implement other types of queries. For example, other queries of interest are streaming queries that I will now talk about for brevity. So we are, of course, kindly invited to check the paper for all the details. So let's start with a preliminary observation. In general, the algorithmic literature is rich of solutions regarding the string dictionary problem, but such solutions are relevant for generic strings, I would say. And by generic, I mean that they are of variable length. Usually they're drawn from a larger alphabets, for example, the ASCII set of characters, and usually they do not feature specific properties to aid compression. Instead, k-mers have all the opposite properties, namely they are of fixed length, equal to k. They are drawn from a very small alphabet, so just for symbols, so we can get by using a 2-bit encoding, and they are extracted consecutively from DNA. So the answer to the question in the slide is yes, if we want to improve the efficiency of our data structure. So in order to obtain a space-efficient solution, we're going to first exploit the following equivalence between a set of k-mers and a de Bruijn graph. I will not really talk about de Bruijn graphs. It's just sufficient to point out that there are efficient software tools that run the following preprocessing flow. So we build the de Bruijn graph starting from a collection of input files that we want to index. Then we compact this de Bruijn graph into the so-called compacted de Bruijn graph. And then if we path cover the compacted de Bruijn graph, we finally output a collection of strings that are usually called the stitched unitics or simpletics. That is a collection of strings without duplicate k-mers. So I'm going to assume from now on that this preprocessing flow has been run from our row collection of files, and that we have this collection of strings with no duplicates as input for our dictionary. So let's now introduce the first ingredient of our solution, the so-called super k-mers. So we have the following empirical property that consecutive k-mers are likely to share the same minimizer. Well, the minimizer is just the smallest substring of length m at most equal to k according to a given order R. So here in the slide, you see an example for k equal 13 and m equal 4. So suppose that AACC is the minimizer, and R in this example is just the simple lexicographic order. So as you can see, all of these k-mers in sequence, they all share the very same minimizer. So I'm going to say that a maximal sequence of consecutive k-mers having the same minimizer is a super k-mer. So in this example, this underlined sequence is the super k-mer. And about super k-mers, I have two important observations. So the first one is actually a consequence of the property of minimizers in that consecutive k-mers are likely to share the same minimizer. So this means that we expect to see far fewer super k-mers than k-mers, which allows us in turn to spend space in our dictionary proportional to the number of super k-mers rather than the number of k-mers, thus sparsifying the dictionary. And the second observation is that a super k-mer of length, for example, S, is a space-efficient representation for the set of its S minus k plus 1 constituent k-mers. So you can see here an example with k equal 13. So for this super k-mer of size 19, it contains 7 k-mers, and we spend 2 times 19, so 38 bits for 7 k-mers, which is about 5.4 bits per k-mer. So which is of course much better than the trivial cost of 26 bits per k-mer, and especially if we preserve, as you can see here on the left, if we preserve the chain between consecutive super k-mers, the space tends to become 2 bits per k-mer, which is optimal, because we avoid to represent twice this shared overlap with this yellow background, this shared overlap of k-1 nucleotides. And so in this example for k equal 13, we go from a trivial cost of 26 bits per k-mer to 5.4 by representing the super k-mers, and then to 3.2 more or less by preserving the chains between the super k-mers. So now the question is, right, but how do we index super k-mers? So we understood that we don't have to break the chains of super k-mers in order to not waste 2 times k-1 bits per super k-mers. So what we do is locate super k-mers in our string collection. That is, we store an array of offsets into the string that indicate the starting position of each super k-mer in the strings. And for fast identification, I would say, of the set of offsets for a given minimizer, we store the offsets in the order given by a minimal perfect hash function built over the set of distinct minimizers. So now, upon lookup for a k-mer G, we locate and scan the bucket of the minimizer R of G, which is the set of super k-mers that have minimizer R. So in this example below, we see that this specific minimizer R has 2 super k-mers. So its bucket has a size 2, and for a query that has minimizer R, I have only to scan these 2 super k-mers. If you don't know what a minimal perfect hash function is, I have this bonus slide to help you. In few words, a minimal perfect hash function is a data structure mapping a set of n distinct keys into n distinct numbers from 0 and n-1. So these data structures in particularly can be realized in just a few bits per key. And I'm going to use the most efficient and also most recent implementation of minimal perfect hashing in this work, which is pthash. So you may also want to check this out. So let's consider now a practical example with 4 strings. As you can see here, for k equal 31 and minimizer length m equal 8. So in this case, we have 285 k-mers and 24 distinct minimizers. As you can see on the right, the offsets of the super k-mers associated to the minimizers, so are actually these ones. And so our data structure looks like this. So we have the minimal perfect hash function f and the array of bucket size stored in prefix sum in order to translate the result of the minimal perfect hash function evaluated over a minimizer into an index into the offsets array. So for this example, KMARG, we know the bucket is going to be of size 2. And in fact, 9 and 255 are the two offsets of the super chimeras that we have to scan. In this example, this particular chimera is found in the second, represented by the blue arrow, in the second super chimera. So that's sparse hashing. Of course, we have a problem here, and the problem is that some buckets can be very, very large in terms of number of super chimeras. And so a query inspecting a large number of super chimeras, of course, will be very slow in practice. But here we can exploit another important property of minimizers to solve this problem. The fact that they are skewly distributed in DNA strings. That is, most of them appear actually once, and very few of them repeat many times. Here you see a concrete example in the following table. So, so this is the distribution of bucket size, namely how many super chimeras we have per bucket. And so you see that there exists a value of m, of minimizer length, from which the distribution is always very skewed. So namely more than, say, for example, 97% of buckets contain just one single super chimera. So of course, we want to take advantage of this empirical property. So to do so, we fix an integer L, and by virtue of the skew distribution, the fraction of buckets having more than 2 to the L super chimeras is small. So we can afford, if this is small, to build another minimal perfect hash function over the set of all the chimeras that belong to such super chimeras. And in particular, we are going to store in an array at the position given by the minimal perfect hash function for a chimera G, the identifier of the super chimera where G is present. So upon lookup, I don't have to scan many super chimeras, but just one single super chimera. And in particular, the super chimera identifier can be written space efficiently, just using few bits. So we group buckets by size, and buckets whose size, say S, is in between 2 consecutive powers of 2 belong to the same group. And we build a minimal perfect hash function for all the chimeras from all the super chimera in the same group. So all the chimeras in group I, for example, have their super chimera identifier written in just I+1 bits. Why? Because we know that a chimera in group I belongs to a bucket containing at most 2 to the I+1 super chimeras. And so its super chimera identifier can be just written in log in base 2 of I+1, which is actually I+1 bits. So that's skew hashing, and so this skew index takes a very small space in practice. So here is an example for L equal 3. So for this query, chimera G, in this example, we retrieve its bucket size. As you can see, it contains 13 super chimeras. So I don't want to scan, in the worst case, 13 super chimeras. In this case, I have that 13 is larger than 2 to the L, which is 8. And so we know that its super chimera ID can be retrieved from the skew index, here represented by this minimal perfect hash function on the right. So since log in base 2 of 13, taking the seal, of course, is a 4, then I know that its super chimera ID can just be written in 4 bits. So that's an example of skew hashing. Now I will illustrate some experimental results on full genomes of the following organisms. So you see we have the full genome of the code of the common kestrel, the human genome, and a large pangenome of bacterials. Here on the top of the slide, you have all the information regarding our experimental setting. So the use processor, we use a GCC compiler with full optimization flags, and all the experiments are single-threaded, and the code is available on my GitHub, and it has been written in C++. So first of all, the tuning of SSH, namely the trade-off obtained by varying the minimizer length, M. So here we have a trade-off because small values of M create longer and fewer superchimeras, which is better for space, of course, but small minimizers have also a higher probability of repeating in the DNA strings. So it means that buckets will be larger on average, thus also increasing the query time. So as a good rule of thumb, you can always select M as given in this formula. So we take a log in base 4 of capital N, which is the total number of nucleotides in the string collections. And so I'm going to use as value of M these values in bold. Next, we consider comparison against competitive approaches, namely the DBG-FM, which is based on the celebrated FM index, and two recent indexes, pufferfish and blight, also based on minimal perfect hashing. So this is the result of the comparison regarding space and lookup time. So on the left, you have the space of the dictionaries, and on the right, the lookup time. So you can actually see, without going through each lines, that our solution, SSH, is more or less one order of magnitude faster, as you can see, that the FM index, for just sometimes 2 times more space. Instead, compared to other schemes based on hashing, we are between 2 up to even 5 times smaller in space, with comparable or even faster query time. And this is also going to be true for streaming queries. So when we are not just performing lookups in order, when we are actually performing lookups in order from a query sequence and not random lookups. So that's also the case in which SSH is sensibly faster than previous schemes based on hashing. Finally, regarding construction time and space, so in this table, I am reporting the number of minutes that is taken by the construction process for the SSH index and the RAM used during construction. So we can see that SSH is very fast to build, although these numbers in these experiments are relative to the in-memory construction. So the entire construction happens in internal memory for SSH, and that's why RAM usage is high, as reported here. So generally, as you can see, twice. I mean, it's much higher than that of blight, usually by a factor of 2, and comparable to that of pufferfish. But this actually has changed in the latest releases of the library. So we're using an external memory algorithm. So to conclude, we presented SSH, an efficient solution to the so-called chimera dictionary problem. And in particular, SSH embodies a good trade-off between space and time. And we used a rich toolbox consisting in spectrum preserving string sets, minimizers, minimal perfect hashing, and also compressed encodings, for example, Elias-Fano. And we explain these two main ingredients, so space indexing of superchimeras to obtain good space effectiveness, and skew hashing to guarantee very fast lookup for large buckets. The code is available again, so please go there and check the code out. And as already mentioned, I added several things to the library, such as the external memory construction, support for compressed abundances, so the chimera counts, so how many times a given chimera repeats in our data set has been discussed in a separate paper. And also I've added support for navigational queries. Okay, so that's all, and I want to thank you for your attention.